Okay, I think that it's about 6 o'clock, so we'll just make a start. So this week, what we have planned is to go through the material as it relates to exam 3. So it's kind of a chance to go through a little bit of revision so that you feel all ready and geared up to tackle the exam. But before that, I do have few announcements to make. Sorry, that's my cat meowing there away. I have few announcements to make, so that'll give a chance for anyone who's a little bit behind joining us to get caught up. So you might have heard me mentioning it earlier on, and I think I actually sent an announcement of that this week on the coming Thursday, it's the last day of the semester to withdraw from a course without you being recorded a letter grade for this course. So what that means, and usually if you're here on this session, you're in a great place and you have nothing to worry. But sometimes things happen and things don't go the way we plan at the very beginning of the semester. We instructors try to go through and check with our students how they are doing, and if it doesn't look like it's working out great, if there's other challenges that are pulling you away from studying or submitting the work, we will come up with strategies to help you to get caught up. And if that doesn't work and you deem that you just have too much going on in life, it might be that this is something that you might want to consider, that is it better to just drop the course and not get a poor letter grade for it? Because every time you get a letter grade, that will have an effect on your GPA unless you go back and resit that class. But really, if that was the case for any student, these would be students who have not handed in any work since the very early weeks of the semester, or who just are missing so much work that it's going to be hard for them to get a good grade. Remember, at the end of the semester, I do give you a few chances also to bring up your grade a little bit if it's not quite where you want it to be. So I guess that what I'm trying to say is that if you're here and you're here today, you're probably doing really well. You're probably looking of a great grade where you're heading. But if for some reason you were here and you know that you haven't been submitting work throughout the weeks, the weekly homeworks, and you haven't been taking the exams, it might be that you're just missing so many points that this is really the time to wrap up the sleeves and contact me and talk about how we can get you caught up so that you're able to get enough points that you can get a decent grade for this course, because nothing's as pointing as getting that bad letter grade there on your transcript that you then kind of have to compensate with other classes and acing them. Anyways, what I'm trying to say is that the onus is on a student to monitor their progress, and student can contact the registry if they feel that it's not working out. I typically try to encourage students to talk with me a little bit. I have a pack of tools that I can pull in and try to help you. I will not just set you up so you can skip all the work that has been updated, but I can help you to try to get caught up on those. However, sometimes we instructors as well may help, and as counterintuitive as it might sound, sometimes it might be better for the student to be dropped from the class than them just hanging on there, not sending in any work, because we really don't want you to have bad letter grades as a final grade for this class. So if I don't hear from a student, I typically reach out to that student and kind of try to connect to make sure that if there's something that I can do to help you and kind of have a bit of an understanding, better understanding what's going on, why you might be struggling on staying on top of the classwork. But these are all steps that we do because we care. Technically, I can, if I don't make the connection with the students, if I don't hear back, if they're not responding, I can also at certain point make the call that it is in the student's best interest to be just dropped from the class. And I don't do this all the time because I do understand that life happens. Sometimes you work a little bit of ahead, sometimes you might need to play a little bit of a catch up, but there are certain points in the semester when I really focus on that, and this week would be one of those weeks. So if it's not working out for you and you're not wanting to reach out to me, then monitor your messages. I might reach out to you and then we can try to figure out what's going on. But if I can't get hold of you in any way, then there is a theoretical chance that you also could be dropped. I rarely drop any students without having a dialogue with them first. So that's what I'm doing this week. So just keep an eye on those messages. If you get a message and you're not responding to that, that might be a concern. But like I said, if you're here today on this class, you're probably doing really, really well. I'm going to give a chance to for anyone to ask any questions if you have about this, but like I said, I don't expect this to apply to most of you. This only applies if for some reason student enrolls to classes, starts to work on class, but then just falls off the radar. Sometimes students think that, oh, I've had it with the college, I'm not going to continue anymore, and they leave, and then a couple of years later they want to come back. Okay, you would still have that bad grade for this course hunting you on your transcript. So that's the reason why we try to make sure that we also clear you off from the transcript if it's not looking good for you. But like I said, I don't expect this to be a concern to anyone. I don't see any messages having came through. So I'm just going to continue and we'll take a chance again to address any questions that you have. I'm going to continue to just kind of give you a really quick run through of this week, what we're going to be doing this week. Only piece of assignment that you really have is completing your exam tree. So as you see on this course, I try to build that you have nothing else but the exam whenever there's the exam, without few exceptions. So you have the whole week to tackle that. And I'm recording this session today with the intention that we're going to go through the study guide, the exam tree. And for that reason, I really have nothing new to share with you on the coming Wednesday. So unless there's any one-on-ones that students ask from me, there will be no formal teaching on the Wednesday. So it will be just for one-on-one meetings and things like that with students who feel that they have gaps in certain areas or so. Make sure that you take the exam, and I've set the due date for the exam on Saturday. So, and at the midnight, so try to do that before that. This exam will cover 3 chapters. We'll talk about metabolism, and then we'll talk about the photosynthesis and cellular respiration. And those 2 last chapters really we kind of tackled as a bundle. So it's not a huge amount of material. And the reason why I have more exams and less content in the exams is that you can still afford to not ace every single exam to 100%. It's still not end of the world. This exam has been scheduled for 2 hours. You get to pick the time and the place where you take that exam. Actually, only 32 questions because there's just really not that much material that we have covered in these 3 chapters. They add up to 80 points, so the same weighing as with the previous exams. You should sit the exam on one go. So once you start, you should finish it within 2 hours. I really don't expect that most of you would even take that long. It is an open book exam, so you can use your notes, you can use your textbook, you can use resources that you have available. It is not proctored. But what's important for me is that you know where to go for the answer if you don't have the answer in front of you. And really remember that this course is really fundamental concepts. You should grasp these so that when you move on to the advanced classes, you're not going to be struggling there. So this is kind of bringing everyone to the same point. And if you have any assignments that you've missed up to this point of the semester, this week would be the time to get caught up on those as well. So if you need anything reopened or want to reattempt an assignment, please contact me and I'll make sure to work with you so we can figure out all that. But at the end of this week, we should be there with all the work sent in, no missing assignments, and the exam tree completed. That would mean that we're about 3/4 through the semester's workload, so that's quite exciting. And moving onwards then for the remaining of the semester, we're really going to shift our focus from these general concepts and looking at the microscopic scale of life, cells. We're going to shift and start looking at larger scale things such as genetics, DNA, how DNA affects in the organism, and so on. So we'll kind of shift a little bit of focus. So that's why I also want to kind of wrap up this set of topics with an exam so that when we move on to the next exam, you have a whole new set of material. Many of you might have seen my announcement where I said that I wasn't quite sure, I didn't have a study guide for this exam yet prepared, and I wasn't quite sure what we're going to do today. Well, I did sit down and I did take the time to prepare a study guide for you. So you will be able to find the study guide just before the lecture exam 3. So we will use that study guide to go through the material for this exam. So it's kind of a chance to review. What you've seen in my study guides is that there's gaps here and there. And whenever there's a gap, that's probably an important term or important concept that I really want you to master. And only time will be showing the answers to those gaps is here and now today. And of course, the recording that I will make available. So I will not be posting a completed study guide anywhere. So if you want to get the answers and you can't figure it out for the study guide, only way to get those is really by either attending today or watching that recording. Of course, if study guide is not your way to learn or study, you can just sit, take your own notes, or you can just listen, whatever learning style works for you. But I just wanted to put it out there. So if you haven't had a chance to open or download or print however you wish to study the study guide, it is there now, and we will start working through it in just a few moments. So this gives you a chance to do so. There is also, I just want to show you the lecture exam with all the criteria that we have been talking earlier on the previous slide. It is underneath that study guide. In general, I try not to do late submissions. If you have missed an exam, do get in touch with me though, and I'm open to discuss and figure out if there's something that that can be done so you can attempt the exam, but that's not automatic. So I just wanted to put that out there as well. So this is really all that we will be doing this week, doing the exam. So that's what I dedicated this week for. In order for me to take the attendance for today, rather than just asking you to post names, I thought that I really should find out how things are working out. So I would ask you to share as little or as much as you feel comfortable with me as your attendance for this week or today. On a scale from 1 to 5, how is this course working out for you? I'm not asking in general life, how is life going, even though you might tell me about life in general if you wish as well. Your answer can be just simply a number. And if you put number 1, I'm assuming that that means that you're pretty stressed out about this course. This course is something that's making you nervous and it's not ideal. And if you post number 5, I'm assuming that you're doing really, really good with this course. You're not worried at all. You feel that it's rolling nicely. And then everything in between with the grades 2, 3, and 4. If you do want to share something else with me, of course, I'll be happy. If you probably post me that you're super stressed, I might be reaching out to you later on just to make sure what we can do to get you away from being super stressed. Of course, whenever you're learning something new, it is a little stressful. I get it that there's a GPA at stake and so on, so you might want to do well with this class. So I'm not expecting that you have to be super chill, but I definitely don't want anyone to be miserable because of this class. So if you can do that for me as an attendance, I greatly appreciate that. So it kind of serves a double purpose. And before I jump in to the study guide and going through it, because I'm looking very closely at the watch, I don't want to run over too much. I just want to take a moment to tackle any questions that might have came up, anything that you might want to ask about anything I've shared so far, or anything that's on your mind. If I have missed a question in your chat, just feel free to unmute yourself, raise your hand, give me a shout out. If not, we're just going to move on in just a second ahead. And I'm seeing the answers on the chat, and I feel good that many of you are not too stressed about this class. I promise that my goal is not to catch you out on something. I'm here really to help you to succeed. So I don't want you to be stressed. I try to make this low stakes. There should be multiple chances to get those points. And remember, even if you're not quite where you want to be, you will be there by the time we finish the course. There will be also chances to get those any points that you might be missing. Well, on that note, I'm just going to jump out of the sharing the screen, and I'm going to share the window where I have the study guide. And I know that sometimes I work through material so fast I miss things, but I believe that at this point you should be seeing a screen of a PDF file for a study guide where it goes through the exam 3 chapters 6 and 8. If you're not seeing that, then I probably have clicked somewhere wrong, and please don't hesitate to just unmute yourself and shout out to me. Oh, it's not a PDF. Let me see. So you should be seeing some sort of a screen. Let me just check that I have uploaded the right file for you as well. So when you go to the, you should be able to download the study guide. I realized that probably the bottom header or letterhead is not quite correct. I finished the study guide late at last night, so I might have overlooked something. But as long as you're able to see my screen for exam 3, we'll just solder on and go through some of the key points of what you should know for the exam. And the first chapter that we had a look of was all about metabolism. And this chapter, I think I shared with you, is often addressed in many different names in different textbooks. It can be dynamic cell or chapter about energy, but really all of these tackle the same things. We're talking about how we take the nutrients from food, convert them through biochemical reactions into energy, and how this energy then can be converted into ATP, which is really the energy currency that your cells use. So pretty much all cell processes are driven with ATP. So that's the big gist of this this chapter and what I'm wanting to cover on it. There's quite a few concepts. So in comparison to the chapters 7 and 8, this is the heaviest chapter in terms of number of definitions, number of concepts. So don't feel overwhelmed. It's not going to be quite as heavy for all of the chapters. So what I did want to start by talking a little bit about the energy. And we used a definition that most textbooks use. I think that especially most high school textbooks kind of tend to stick to this definition that energy is an ability to do work. And there's nothing really as such wrong with that definition. Yes, energy can take many different forms, and it kind of highlights that energy is just something that can drive reactions, can drive work to take place, but there is a little bit of a oversight in that definition. I would say that it's a little bit of an oversimplification, because even though that energy wouldn't be available to do work, it doesn't stop it from being an energy, if that makes sense. So even if the energy is not currently available, if it's stored in a form or in a manner that we cannot immediately access, it doesn't mean that it wouldn't be energy. It is still energy. It's just in a form that's not accessible. So that basic definition that we saw here kind of overlooks that. So that's why I wanted to show it there. And I think that in my homework questions and in my exam, you probably already picked it on that a lot of the exam questions are questions from homework. So if you have worked through your homework, you should have seen a lot of the questions that you face in an exam. It's not a definite one-to-one ratio, but I try not to really do curveballs. But in those questions, I could probably ask for both of these definitions. I could ask you to spot that, yes, energy can be defined as an ability to do work, but I would also want you to be comfortable pointing out that, well, this is the definition that we use, but that definition even has some flaws. And then going on to the fact that we saw here that there is still energy, even though it might not be available at that very moment. Another thing that we saw when we discussed about the energy is that ultimately all energy on this earth originates from the sun. So energy can be changed to various different forms, but all of these more or less are from the sun originally through one or more conversion processes. And as such, the energy really passes through the system as we use it. So it can change shapes, it can be changed to different forms, but as it passes through the system, we do not get energy appearing out of the blue. It does not appear just out of nowhere. And similarly, energy doesn't disappear just somewhere. We can trace where the energy is lost. So energy can change forms, and that's part of the process as we see that energy passes through this system, but every time that we change the form of the energy from one form to another, a little bit of it is lost in this process. So it's never 100% of the initial energy that we get when we change forms. And the most typical form how we lose some of the energy is as heat. So I think that the example that I used earlier on our classes was that I spoke about how you fuel up your car with gas. And that gas then gets converted into energy that helps us to push that car ahead. But you don't get quite as much out of that energy as we had bound to the gas. So there's some of it lost in this conversion process. And especially in a car, we see that very clearly that that's lost as heat. But the same is for any other conversion. We always end up losing a little bit of the energy. And the heat, like I said, was the common way how we lose it. So I hope that that makes sense. If at any point I say something that doesn't make sense or you have further questions, please do not hesitate just unmuting yourself, giving a shout out. Okay, I hear, sorry, I only see now that you do not have a study guide. So if you go to the, I'm gonna stop sharing this for a second and I'm gonna share my screen completely. So if you go to the Blackboard course that we have, and then you look under the content area. So this is the area where you would typically land on, and you scroll down there to week 10. I don't have a specific learning module for week 10. Instead, there I have exam, and on top of that I have the study guide for the exam. Once you click on that, that should open you a page, and then you should be able to click on. My view is a little different from students.
Can I show you what I have, because my study guide doesn't look like that.
Let's have a look.
Okay.
Do others have the same? It might be that I've uploaded something wrong. Let's have a look. Study guide, exam 3. But I would expect that the study guide is missing some parts, but it's always possible that I've uploaded something wrong late at night. So this is what you should be able to see when you download that study guide. Are you able to see that?
No, I have like just I don't know, it's like a bunch of words on one sheet. It doesn't look.
Let's tackle this. Does anyone else have the same problem? If someone else has the same problem, just please drop a message to the chat or let me know, and we can tackle that. If no one else has the same problem, what I think I can do, I can just send you the study guide through the Blackboard messages. So if you just check your messages in just a moment, you should have the study guide there. Just bear with me as I upload it. I'm just going through my files. Sorry, I'm clicking away as fast as I can. So now if you check your messages on the Blackboard, you should have received the study guide there from me. I'm not seeing that anyone else would have the same problem. If you do have the same problem, please let me know. If you're opening the PDF and it comes across as corrupted or something, make sure you have a PDF reader. If still it's not working out, do let me know and I'll definitely try to help you. But since it looks like the majority of the class has this study guide, I'm just going to jump back on and we're going to continue working through it. I will work with you if even now the study guide doesn't open up the way you're expecting it to open up, and we'll figure out what on earth is going on there. Like I said, you should be in a version where you are just missing everything that's written on the blue. Yes?
I was going to say, Brittany, if you have a phone, maybe you could take pictures of the notes.
That's a great idea. Thank you for sharing that.
Yeah, thank you.
I'm sorry, I will stay behind Brittany and figure it out with you, but just in the interest of time so that everyone else gets the answers, I'm gonna keep working through this, and I don't want you to feel that I'm ignoring you. I'll just have to do a little bit more troubleshooting to figure out what's going on. So let's have a look at the next portion. And in the next portion, now that we've talked about the energy, I wanted to talk a little bit about nutrients. So most of us have covered, I believe, in your high school classes or elsewhere, the concept that nutrients are recycled time after time, whereas the energy really is kind of more of one shot through the system. So that's the big difference. If you look at this diagram, you might have seen it when we talked about it in the lecture video. You have nutrients that pass through the system at various different levels, but we never really run out of them because we can reuse them time and time and time again. So there were three roles that we can in broad give to various different organisms in an ecosystem. And the first role is that we can classify them as producers. Producers are organisms that are able to do photosynthesis, so they make food and energy out of just the sunlight and oxygen. And that's what they do. They are able to convert that solar energy into something that can be then stored in a chemical format, usually as sugars. Well, most plants are producers, and we'll talk about the process of photosynthesis in just a little bit in the next chapter, a little more. Well, most organisms that are similar to us humans, we cannot do photosynthesis. So we rely on using the producers or other organisms that have used producers to get our nutrients. So we are classified as consumers. So we use other organisms to gain the energy that they have stored from the sun. And then we have the third class, which is the decomposers. They break down organic waste and release those molecules, those elements to be used again in this cycle. So you really see the difference between how energy passes through the system. It goes through the system and then it's gone. Whereas with the nutrients, these nutrients get reused time and time and time again. So I hope that makes sense. Really, I'm not hoping to go too much into about nutrients. I just really wanted to highlight that that difference between how energy versus nutrients pass through the system. Well, at this point, we get to the really meat of the chapter, if you wish, the most important topics. So we're going to start talking about metabolism, and there's no better way to talk about it than to define it. So metabolism refers to chemical reactions that living organisms have to do to stay alive. And really, it refers to all of those chemical reactions. So that's the key word I left out from the student version of the study guide. So all chemical reactions that help you to stay alive are part of your metabolism. So that's often a term that students think is a lot more complex than what it is. Keep it simple, stick with these basic definitions, and you'll do just fine. Well, if metabolism referred to all of these chemical reactions, then a metabolic pathway would refer to a series of connected chemical reactions. So this would be, and I think we saw this diagram that I have put here on a tiny scale. This related that when we had one chemical reaction that was needed for the next chemical reaction to happen, which then gave rise to the following chemical reaction, and so on and so on and so on. So most of the time, chemical reactions don't happen or exist by their own independently. They are part of a chain of interrelated reactions, and these chains of interrelated reactions would be our metabolic pathways. There were two main types of reactions that I wanted us to know about, and these were our anabolic and catabolic reactions. So we're going to talk about them just briefly. And the anabolic reactions refer to when we take individual small either elements or tiny molecules, and we use those to build something larger, such as this molecule, large molecule that we see as a result of this reaction. However, what we find most of the time is that for anabolic reactions to happen, they require energy. So putting together these individual small elements or molecules to make something that makes a larger molecule, that requires effort, that requires energy. Sorry, I've got flies flying around here. I'm not just waving the air. So that's our anabolic reactions. So making large molecules that consume energy. So this is really what your body does. For example, when you're growing up, your body is developing, you're growing new parts of the body, or your original parts are growing bigger, more complex, so on, so on. All that takes energy. Catabolic reactions instead refer to when we take this large molecule that we just discussed about, and we break it apart into smaller either elements or molecules. Well, this process happens relatively spontaneously. Most things in life tend to break apart over time. And as this large molecule breaks apart, it also releases some of the energy. So if you think of the food that you eat, and your digestive system breaks this food into smaller and smaller and smaller molecules, as your digestive system does that, it releases with each step more and more energy that your body can then use to drive the processes. So that's why we end up finding is that our body kind of goes between these two types of broad categories of reactions. So it is important that you can tell them apart from each other, and you can kind of understand that catabolic reactions are probably easier to happen. They happen more naturally because it really doesn't take any effort for larger things to fall apart into smaller things. But as they fall apart into smaller things, we are left with free energy that we can then use for other purposes. I hope that those two make sense. If not, let me know and we'll kind of look at them in a different way. But what I went next to talk about in the study guide was about different types of energy. And I've listed only some here. So there's going to be a bunch of other types of energy that you'll see. And out of all of these, there were really two that I wanted us to focus on. I wanted us to talk about kinetic and potential energy. And if we look at the kinetic energy to start with, one of the definitions how kinetic energy is often defined as energy in motion. So whether it's this pen or whether it's a baseball ball that I throw, and as it flies through the air, that object has now kinetic energy. So it's energy that's in the motion. And better way probably to understand it is by comparing kinetic energy against potential energy. Well, potential energy is energy that's stored in an object. The energy is not really being used to make something happen, like flying off that object at that very moment. It's just potentially there, and it can be used later on when needed. So potential energy really refers to stored energy. And I think we used an example of carrying a washer to a high-rise building, or when you put books on a bookshelf. All of those objects, as you move them further away from the Earth's surface, they all now store some energy. And this energy can be released as these objects, for example, would fall to the surface of the Earth because of the gravity. So they have this potential to do this movement, but that movement doesn't happen yet. So that energy is just stored there. I hope that makes sense. Again, I realize that now when we're going through just a kind of a review of these, we're going through them rather quickly, spending less time on examples. So if something feels odd, let me know and we'll kind of zoom into it a little more. But what I really want to do, I want to go through all of the key concepts at least. Then we talked about measuring energy, and there were a few different terms that I introduced. I introduced the term of joule. That's work done by the force of 1 Newton, but that's not probably the most useful measure of energy that we're going to be looking on this class. The measure of energy that I'm really interested for us to pay attention is talking about calories. And during the class, we defined or found a definition for what is a calorie. And what we saw that it was the amount of energy needed to raise the temperature of 1 kilogram of water by 1 Celsius degree. So a certain amount of water gets warmer when a calorie is released. And this definition seemed a little vague, at least to me, until we took time to figure out how could we measure calories. And a lot of the time when we measure calories, we talk about calories, you see food products, and on those food products you got these labels that tell that how many calories or more kilocalories these things that we can eat contain. Well, they didn't just come up with that number out of the thin air. There is an actual way of measuring the amount of calories that you can have. And the method for doing that was by using a tool known as bomb calorimeter. And bomb calorimeter really relies on the concept that we take an object, in this case the piece of food, and we burn it in a container. And this container, we direct a spark in there. This container is rich in oxygen. It's ideal for burning that stuff. And as the material burns inside the container, it releases heat. And we're going to capture that heat by putting this container inside another container that's filled with water. And then by ensuring that the water gets mixed by having some kind of a stirrer or propeller in there, and having also a thermometer, we can tell about how much the temperature of the water has changed as we burned that piece of food or piece of material in there. So this was really a way that how we can do them backwards calculations to figure out how much energy was released by burning that object. And that tells us that how many calories that object contained. So I believe that we in the class actually drew a bomb calorimeter together. So I've just provided a simple diagram here, but I hope it helps to kind of remind you of that concept. The next thing that we talked in the class was a concept of energy of activation or activation energy. And we came back to this in some of our labs later on. So it was kind of a way to get us thinking of something, and then we'll return to that to really tackle the fine details of it. What you'll see typically in diagrams is that, and I'm just trying to get my tools, is that on x-axis, we typically have time. So as time moves on, we move along on the x-axis. And on y-axis on this diagram, we're seeing the amount of free energy that's available. So what we have in this diagram is some sort of a reaction that's starting to take place, and it runs out of energy and it stops. Another important thing that I want to highlight from this diagram is this threshold that I've drawn here with dotted line. This threshold was called activation threshold. So if the amount of free energy reaches this activation threshold, then the reaction will move forward. If it does not reach this activation threshold, the reaction simply stops from happening. And in this first example, of course, the activation threshold was not met, so there was no reaction taking place. On the second diagram, now we are actually going to meet this activation threshold. So the amount of energy for a reaction to start is reached. And as we reach this activation threshold here, now this reaction is going to keep going forward and taking place. So if the activation threshold is met, the reaction proceeds. Well, these two, I think, are intuitively quite clear. Most people are able to grasp that. Even though it's a little vague, it makes somewhat sense. Well, this is where it really starts to make more of a sense when we can apply a little bit more information to what we've seen before. So far we've seen the activation threshold, which was represented by this green line. And what we saw that the reaction could either meet the activation threshold and the reaction proceeded, or it could be that it did not meet the activation threshold, and in that case, the reaction did not proceed. Well, here comes the interesting part. We can take something like, for example, an enzyme, since we've spoken a lot about enzymes on this course, so let's keep talking. And this enzyme can help us to bring down the activation threshold from its initial value to a lower value. Now, if we have brought down the activation threshold, it's going to be easier for us to meet that threshold and for the reaction to take place. So you saw that initial curve that I had at the first diagram, which was initially not enough to trigger a reaction to happen. But now that we have brought down the activation threshold, it actually is. So lowering the activation threshold can help us to make sure that the reaction occurs more easily than what we saw before. And many enzymes actually do this very same thing. So many enzymes help us to lower the activation threshold. Of course, you remember that enzymes could be doing a bunch of other stuff as well, but one of the things that they do, they could help by lowering the activation threshold. Any questions about this concept? I know that it's a little abstract, and that's why I really think that doing the labs helps a great deal. And I think that we saw some of these earlier on. I really would feel that it'd be much more useful if we could do hands-on labs. And we are actually developing lab kits that you could use then at home to tackle these labs. But we're not quite there yet. So that's something we're working. Well, I'm not hearing any desperate bleeds of, oh, I'm totally confused, what's going on. So in that case, I'm going to keep working through this PDF a little longer, a little further. And one of the things that I spoke to you about was laws of thermodynamics. And actually, there are several, but we're just going to focus on the two first laws of thermodynamics. And when we talk about this, it might sound very intimidating, but really both of these laws of thermodynamics are very intuitive, and they tie very closely with what we saw when we first started talking about energy here at the very first page of the study guide. So they're basically just replacing what we already saw. So let's have a look through two laws of thermodynamics. First of all, the first law of thermodynamics says that energy is conserved. Well, what that means is that the amount of energy in the universe is constant. So energy doesn't just appear out of the thin air, or it just doesn't disappear. So we can always follow that energy as if it was coming from somewhere and it is going to be going somewhere. So that basically just summarizes that very concept that we talked about earlier on. The other thing that we saw was that energy could be either transferred or it could be changed. So changing energy from one form to another is kind of easy to get your head around. For example, plants change the solar energy into sugars. And then we humans can consume those sugars and change that energy into ATP. But every time that we do that, we change the energy from one form to another, we lose some of it. And it was the heat that we normally lose some of that energy. Same goes for when we're transferring energy. So you can't just take a super long electric cord and run energy from one end of it to another end and expect that there's going to be the same amount at both ends. If the energy cord is too long, we will keep slowly losing some of the energy throughout the process. So that's what I'm referring to when I say that when we're transferring energy, we're going to lose some of it as well. So I hope that both of those rules or laws of thermodynamics actually make sense when you reflect on everything that we have discussed so far. And we're just going to continue in a second. We've got a very attention-seeking cat around. The next concept that I talked about with you was the concept of entropy. And the term of entropy referred to the relative amount of disorganization. So relative amount of things not being on their original or on their ideal places. And I think the easiest way to think about entropy and the concept that we can have a lot of entropy or we can have a little entropy is by using an example. And I use this example of a room in our class. So a room that's very, very, very organized, where everything's on its place. That room is going to have a very little disorganization. So that room is going to have a very little entropy. in it. So there's not much chaos, there's not much disorganization in that room. However, if we looked at this second room where everything's all over the place, it has high amount of disorganization. So that means that it has a high amount of entropy as well. So entropy really refers to the amount of disorganization. And I think that that really covers the key points of what I wanted to say about the entropy. Of course, we see that in nature, it's more likely for things to get disorganized. And that goes back to the concept that we talked earlier on when we talked about the catabolic reactions, how by breaking a large molecule into smaller molecules, we're releasing energy. So this tends to happen spontaneously, and we are left with more entropy as a result of that. We didn't really connect these two concepts in our class, and I'm not expecting you to be able to draw that link in a perfect detail. I'm just wanting to show really at this point that the concepts that we talk about are not individual loose concepts. They all are related, even though the connections might not be obvious at this stage yet. But as you continue your studies, they will become more and more clear how they are connected. One of the final concepts from this chapter that I want us to look at relates to ATP. So ATP is abbreviation for adenosine triphosphate. What adenosine triphosphate is, it's often described as the energy currency of a cell. So cells use ATP, which is just a chemical format of energy, to drive nearly all of their cellular activities. So ATP is going to be something that you keep seeing time and time again as you continue your studies in human physiology or physiology of other animals, and so on and so on. What ATP is, well, we're going to have a look of that, and the name really explains what's there. So we have an adenosine and we have a ribose that form the kind of backbone of ATP. And then we have 3 phosphate groups in addition to that. And that's where the term tri comes. 3 phosphate groups. So that's the energy and the energy currency, the molecule, how we store energy in the cells and what we use to drive reactions in the cells. Well, if we lose one of these phosphate groups, we're probably releasing some energy, and we are left with only 2 phosphates, so di. So adenosine diphosphate, ADP, is a form of what we saw earlier on as ATP, but with only 2 phosphate groups present. So most of the time, what we end up doing when we are storing energy is that we convert ADPs into ATPs. And then when we release energy, those ATPs are converted into ADPs. I hope that that makes sense. That's really to summarize the key concept from what we discussed about ATP. At this point, there's more to the story, but we're going to stick to this level of the detail at this point. I think that the final concept that we talked about in this class was the concept of an enzyme. And we saw enzyme, we talked about that in quite a few labs. It's a concept that keeps coming up and up and up time after time. So it tells you that it's an important concept. So enzymes can be defined as things that assist in chemical reactions. So these chemical reactions would happen anyways, but by adding an enzyme, we might make the reaction quicker, or we might lower the activation threshold. We might make the reaction more efficient. We might make the reaction take place when it normally wouldn't, and so on and so on. So enzymes, we don't define what kind of assistance they give, but simply that they assist in chemical reactions. And I've listed a little bit more detail about enzymes below there. I provided that diagram that I've shown you before, and I refer you back to those lecture videos if you're totally lost about what is an enzyme. One type of an enzyme that we have talked about is a catalyst. This is a special type of enzyme, so not all enzymes are going to be catalysts, but some of the enzymes are going to be catalysts. And what catalysts do, they speed up reactions. They make reactions happen at a quicker rate. And we used, for example, in our lab, baker's yeast as a catalyst solution that speeded up reactions of what we were studying in the lab. So that really wraps up what I wanted to say about that first chapter. And what we have left is two more chapters: the photosynthesis, which was chapter 8, and cellular respiration, which was chapter 7. And I know that it's a little confusing that I'm going to go through them in almost like in an opposite order, but I really think it makes more sense to start with photosynthesis and then discuss cellular respiration, because this is the order how the energy would pass through the system based on what we have discussed before. And we're going to go through these two chapters in a little bit of a faster rate because I don't want to keep you over time. I know that we got a little sidetracked there earlier on with a few things. So I want to make sure that I'm respectful of your time and I'm not keeping you too much of an overtime. If you do have to leave, I totally get it. And there's going to be a recording, this recording made available for you later on. But let's start by looking at what photosynthesis is. And really, you know that I love talking about the etymology of the words, where the words come from. And photosynthesis, really, if you translate it word for word, it talks about putting together things with the help of light. So what we do in photosynthesis, we convert solar energy into sugars. So we take individual elements or individual small molecules and we make sugar molecules, these larger molecules, out of these individual elements. So we're putting together something with the help of the light. And there are many photosynthetic organisms. The ones that we typically think about are plants. So any plants that have green are able to do photosynthesis. Also some algae and some bacteria are able to do photosynthesis. But really it's going to be largely the plants that we're going to be thinking about when we think about photosynthesis. And the chemical formula for photosynthesis that we see is that we have our, and I'm going to just try to change the color of my marker. We have our water. So plants do require water. The other thing that plants are able to use, which is really convenient for us, more of a kind of a good for us kind of a thing, plants are able to bind carbon dioxide. And we know that that's why if we lose all the plants from the earth, we are going to be having trouble with the greenhouse effect, because carbon dioxide speeds up the greenhouse effect and we need to bind it somehow. And there's no better way to bind it than with the plants. Well, by having water and binding carbon dioxide, we still need to have also the solar energy. So the energy from the sun. And plants are able to convert that solar energy with the water and with the carbon dioxide to make sugars. And these sugars, there's many different kinds of sugars, as you might remember. Oh, I didn't mean to click on that. As you might remember from our initial chapters where we had a look of macromolecules, some of the most common molecules that we have. But the typical one that we use most of the time when we discuss about about sugars is going to be glucose. That's just a simple formula, and that's something that's just commonly used to describe the process of photosynthesis. So we're able to convert solar energy together with water and by binding carbon dioxide to make sugars. And in this process, as a byproduct, but as a byproduct that's going to be very useful for us, plants end up producing also oxygen. So really, that's not the original goal of the plants to produce oxygen or bind carbon dioxide. That just happens in the process of photosynthesis. But both of these are very, very useful things for us. So that's the photosynthesis in a nutshell. Would I expect you to memorize how to write each of these formulas? Probably not. Would I expect you to be able to recognize elements of this formula? Probably yes. So I hope that that helps you to kind of think of to what level there is an expectation of the material. And what I want to talk next is about the structure that really does the photosynthesis in a cell. And this structure is going to be our chloroplasts. So we're going to be focusing on that a little bit. They really are the site of the photosynthesis in the cell. And what we find when we look at the chloroplasts are that they have double membrane, so two-layered membrane that enclose all of the structures inside the chloroplast. And what we find that there were these disc-like structures inside a chloroplast. Often they were piled into these stacks known as granums. And these disc-like structures, they were called thylakoids, and they contain a pigment known as chlorophyll. And this chlorophyll is really now what allows for the photosynthesis to happen. A couple of other things that we see inside a chloroplast, that's kind of an aqueous, watery fluid inside there, that's going to be called stroma. So when there's all the empty space that's filled with this watery fluid, that's stroma. So those are some of the structures that I would expect you to know out of the chloroplasts. And then what we moved our attention after that was to talk a little bit about solar energy. Simply because it's so important concept, it ties in nicely here, and I want you to be aware of at least some of the basics of it. And really, electromagnetic radiation contains a large range of different kinds of frequencies. And the one that we're going to be interested in is, of course, going to be the section where we see visible light. But if you look at this little diagram that I have here, you'll notice that the visible light represents only a very small portion of all the electromagnetic radiation that we have. There's going to be a lot of other kinds of radiation that you might have heard about, such as X-rays or UV rays, infrared rays, or even radio waves. Human eye is adapted on seeing the range of the visible light. So that's what we're going to be focusing on. And visible light really can be divided into different wavelengths. So when we talk about a wavelength of electromagnetic radiation, what we see is that electromagnetic radiation is kind of like this wave, and the distance from one crest to another tells us the wavelength of that particular portion of this electromagnetic radiation. And what we'll see that the colors with the largest wavelength tend to be on the red end of the scale, and the colors with the shortest wavelength tend to be on the bluish violet range of the scale. So I listed there for you the different colors based on their wavelength. And that would be something that I think would be very useful, at least to have as a resource that you can refer back to later on. So you know that going from the largest to the shortest wavelength, it's going to be red, oranges, yellows, greens, blues, indigos, and then violets. And mixtures of those then cover all of the possible colors that we can see on Earth and also outside the Earth. So I think that that really summarizes everything that I had for chapter 8, and that leaves us to talk just briefly about the chapter 7. And I know that we have about 5 minutes left, so that's why, if it's okay, I'm just gonna soldier on and kind of cover this, and then we can take time if there are any questions about this or any other chapter. So what we learned earlier on was that all cells needed ATP to drive their processes. But what we saw is that ATP isn't just floating around. What we normally have is that we have sugars. These sugars are produced by our photosynthetic organisms, and then we convert these sugars into ATP inside the cell. So that's the process of cellular respiration that we'll be looking on this chapter. And again, I have described that process here with a formula. So we're going to look at what goes into this reaction and what comes out. So what we see here, we definitely recognize our good old friend glucose, so sugars. And many animals, many other organisms consume other animals to get their sugars from the nutrition. Of course, plants can make their own sugars, so they don't need to be consuming other organisms. They're photosynthetic organisms, so they have both chloroplasts and mitochondria, which is where the cellular respiration happens. Whereas as humans, we won't have any chloroplasts, but we have mitochondria that help us to convert the sugars into ATP. Well, other thing that you need in this reaction is going to be water. And that's why we need to have both glucose and water available for this reaction for us to make ATP. And you remember ATP was the cellular energy currency, as we just discussed. What we are left as a byproduct of this reaction is that we are left with carbon dioxide and some water. And I noticed that I've made an error here. I was meaning to say that we, of course, need oxygen. Oxygen going to the reaction. So humans need to get two things. We need to get oxygen and we need to get sugars. And those we can convert into energy. And as a byproduct, we're left with carbon dioxide and water. And we need to get rid of this carbon dioxide and water. And how we get rid of those, that's the story that we'll discuss on BIO182. When we talk about respiration, so your lungs and that whole system, and we talk about urination. But at this point, what I need you to know is that we just convert sugars with the oxygen to make ATP. And like I already alluded, this process takes place in a cell structure known as mitochondria. And we had a look of mitochondria a little bit earlier on, but now we're going to add a little bit more detail to that discussion. So for the longest of time, mitochondria were thought to be the only part of the cell where ATP was produced. Well, it has now turned out that ATP is made in other parts of the cell as well, but still majority of the ATP is made in mitochondria. So really the role of the mitochondria is to convert sugars into ATP. If we look at the structure of the mitochondria as an organism, we see something very similar. We see double membrane structure, so two membranes surrounding this structure. The other thing that we see that inside mitochondria, we have these folds These are known as cristae. We also discussed about a few other interesting characteristics of the mitochondria, such as that it only contains maternal DNA, and that had few effects on the forensic work. But we're not going to worry about that on this chapter. Well, there's going to be two processes that are closely involved when we talk about the cellular respiration. And these two processes are our oxidation and reduction. So let's have a look of what each of these processes refer to. In oxidation, we remove hydrogen atoms, and in reduction, we add hydrogen atoms. So that's the level of detail that I'm expecting you to know at this point about these two important processes. And these processes are something that you will become more familiar again as you continue your studies. But at this point, what's important for me is that you have the basic idea of the basic definition, what both of these mean. Let's have a look of the glucose breakdown. I have been thinking, I think that this might have been the handout that one of the students referred to that I mentioned in one of my lecture videos. So the process of converting sugar into ATP follows this four-step process. We can skip some of the steps if we need to, but normally we go through these 4 steps. And I think that your lecture video might show a little more detail of that, but really the steps, what you need to know about it is that with each step, we are either generating more ATP or we're generating molecules that can be then later on converted into ATP. So for each glucose molecule that we take, we can get at maximum in ideal conditions 38 ATPs out of for each glucose. But this is assuming that there's going to be oxygen available for this process. So in an ideal condition, we get 38 ATP molecules out of one single glucose molecule. So that's a very efficient conversion. We have very little energy lost in this conversion. Well, things are not always that ideal. Sometimes we do not have oxygen available. And if we do not have oxygen available, we talk about anaerobic reactions. These are also known as fermentation. So in fermentation reactions, we're still taking glucose molecules and we're still making ATP out of those glucose. However, this process will not be as efficient. In fact, we'll only get 2 ATP molecules out of for each one glucose molecule that goes into the reaction. So it's much, much, much less efficient. So it's not really excellent in that sense. The other negative downside of the fermentation processes is that as we do fermentation in our body, we are left with toxic substances as products of this conversion process. So runners especially know that when you run for a long time or when you exercise for a long time, the buildup of lactic acid in your muscles. Well, this is because now your muscles have been pushed to the extreme. They have not been receiving all of the oxygen that they need for converting glucose into ATP. This can be because we just simply cannot breathe fast enough, or our circulatory system cannot quite keep up with the need, or whatever the reason is, we're not getting quite as much oxygen as we would need to convert glucose into ATP. And of course, ATP is what's going to drive then the muscle work of, for example, running around. Well, if we're not getting enough oxygen, not only we make less ATP, but we end up with this buildup of lactic acid. That's a good example of toxic products. It will not do harm initially, but we need time to recover from it. So later on, we kind of pay the price for having done fermentation. So you might wonder, why on earth would we do then fermentation? Well, first of all, it's a very fast process. So if we don't have time for all of these steps that we saw earlier on for the phases of glucose breakdown, the process of fermentation cuts most of these steps away. In addition, another thing that it does, it helps our body to keep going, even if we would otherwise have to stop because we're not getting enough oxygen. So it's a way for us to overcome the limits of our body, limits of our respiratory system for getting oxygen in, the limits of our cardiovascular system for transporting the oxygen to the tissues, and limits of our cellular metabolism of doing the cellular respiration in full oxygen manner. So fermentation can be both positive and a negative thing. Of course, we know that humans have used the process of fermentation even before we really fully understood what was happening there. For example, we use that in the production of alcohol for many beverages, and also production of carbon dioxide, those bubbles in many drinks, and in certain food production processes. So fermented foods have been something that has been discussed largely in terms of whether they're beneficial for the health. And finally, what I have here, and we really didn't have a chance to go into too much of a detail of that, but most of the classes that we have, and most of the classes that I teach, we discuss about the metabolic pathways by using the carbohydrates, especially the glucose, the sugars as an example. But that's not the only kind of things that we can use and only pathways that we find. Of course, we can use proteins to get energy out of. We can use fats, oils, lipids to get energy out of those as well. So there are alternative metabolic pathways to the ones that we typically teach for you. So I hope that that makes sense. That really comes to the very end of the study guide and everything that I had planned to cover with you. I think that pretty much every single question in the exam has been covered on this study guide. I am going to be honest, it's not going to be word for word, especially since I wrote this study guide last night. It's not going to be exactly just find this question and you will have an answer there. You might need to start doing a little bit more independent thinking, applying, but there's no hard curveballs. And remember, you have quite a bit of time on this exam, so it shouldn't be a stressful thing to work through. Of course, as always, I might have made oversights. Sometimes I make mistakes, like you saw some of the terminology there. I also I'm human and I do errors. So if there's anything that doesn't make sense, ask me, because it's more likely that I've just phrased something poorly or I've skipped through something, and that's why you're wondering that what's going on there. But really, that completes everything I had planned for this review session that we have together today. I hope that it has been helpful. As always, there's going to be the recording made available for you. And please, please, please make sure you tackle the exam during this week so you get the points that you so deserve for doing that. And keep in mind that at the end of this week, you really have done almost 3/4 of the course. So the finish line is there on the side. If you don't have any questions, that's it for today. If you have questions, I am going to be around and you just let me know and I will answer any of those. I will definitely go back to the first page, and what I asked just before you log out, if you can drop your name to the chat, I'll take the attendance because we ran a little over, so I want to make sure that anyone who stayed the entire time has a chance to get attendance for that. But that's it for tonight. I hope you have a good night, and thank you again for sticking around. And I'll be here if you have any questions. But otherwise, good night. Thank you. Good night. Yeah, I'm really sorry, Brittany. I don't know what happened that the file doesn't open nicely for you. Thank you, Erica, that the file didn't open nicely for you. If it's any help, I'm happy to try to troubleshoot it with you. I recommend that you try on a laptop or desktop. Sometimes that might help. Sometimes you might need to download a PDF reader. But if there's anything, good night. If there's anything that I can do to help, let me know. And if you need to see any other pages, because I know we went through a little bit before you got a chance to tell me that it didn't open for you, just let me know and I'll scroll through.
Yeah, the one that you sent me through the message worked. Some reason.
Oh, excellent.
Yeah, so
Yeah,
I have to print it out. I just missed the first page. Everything else I have. So thank you very much.
Excellent. Well, I'm really sorry about that. That's probably something that's just computers have ruined, but I really am glad you were able to get to that. And if anything like that ever happens again, just let me know. I really don't want you to struggle because of IT issues.
Okay. Thank you.
Okay, well, if you got all of those, I hope you have a good night. Let me know if there's anything else. And like I said, I will also post a recording of this class.
All right. Yep, I have everything. Good night.
Okay, good night. And at this point for the recording, I will just stop the recording and I will be around as I take the attendance. If you have any questions, please don't hesitate to ask. And as always, you can always as well message me on the Blackboard. But this concludes our session today. Have a good night. Thank you. Bye.